Okay, now let's discuss nucleic acids. Okay, when we say nucleic acids, before it was initially found that uh, it they are in a cell nuclei. Kaya nga yung pangalan niyang nucleic acid kasi nga akala nila noon it could only be found inside the nucleus. Okay? However, with the technologies and discoveries, it was later found out that these nucleic acids could also be found all throughout the cell. Okay, so there are two types of nucleic acids. We have the DNA and RNA. So DNA stands for deoxy, so that's the D. Ribo, sorry, that's deoxy is the D. Deoxyribo is the D. Nucleic acid is the Na. Okay, so deoxyribonucleic acid, and the RNA would be the ribo, and then the nucleic acid. Okay, so for DNA, they are nearly all DNA are found within the cell nucleus, and their function is for the storage and transfer of genetic information, which is actually passed from the existing cells to the new cells during the cell division. While as for ribonucleic acid, this occurs in all parts of the cell. Okay, so if the DNA is with nearly all of the DNA are within the cell nucleus, majority of the DNA are in the cell nucleus. For the RNA, they are all over the parts of the cell, and their primary function is for protein synthesis. So ulitin natin, DNA is for the storage and transfer of genetic information, and RNA is used during protein synthesis. When we say nucleic acid, their structure is actually unbranched. Okay? And they are polymer, at ang kanilang repeating units or yung tinatawag nating monomer unit is the nucleotide. So for it to be a nucleic acid, so merong dapat nucleotide 1 connected to nucleotide 2 and then nucleotide 3 and so on. But these nucleotides should be arranged in a straight chain manner such that they should not be branched. Okay? So, dapat unbranched siya. Okay. When we say nucleotide, so we know that that's the um, repeating unit or the building blocks of a nucleic acid. It is composed of three subunit molecules. So, meron siyang phosphate, sugar, and base. And ganito dapat yung kanyang connections. The sugar holds both the phosphate and the base. So, si phosphate nakakonect kay sugar and si base nakakonect siya kay sugar. The bases and the phosphate are not connected to each other. Okay? So, we have the pentose sugar. Pentose, alam natin, pent represents 5 carbon atom. So, that's a 5 carbon atom sugar which is bonded to the phosphate group and a nitrogen containing heterocyclic base. Okay, so isa-isahin natin ang kanilang structure. For pentose sugar, it could either be a ribose or a deoxyribose. Siyempre, alam natin that the ribose is the sugar for RNA and deoxyribose is the pentose sugar for DNA. Nagkaiba lang sila ng structure dito sa second carbon atom. Okay? For ribose, it has the hydroxyl group on the second prime of its pentose, while as itong si deoxyribose, kasi nga ayan nakalagay, deoxy. Deoxy, ibig sabihin na tanggal yung oxygen dun sa hydroxyl. Okay, so yan lang yung pagkakaiba ng sugar, pentose sugar ng DNA and RNA. One oxygen only. Okay, and always bear in mind that yung difference nila occurs at carbon number 2. Okay, in the pentose. Now, for the base naman, the second component of these nucleotides, so these are the nitrogen-containing heterocyclic base. Okay, so nitrogen-containing, alam natin that the cyclic organic compounds, usually ang component lang yan ay carbon atoms, just like the cycloalkenes or just like the pentose a while ago, puro carbon atom lang, no? But when we say heterocyclic, so heterogeneous na siya, hindi na siya purely carbon atom. So makikita natin dito sa structure that itong thymine, meron ng nitrogen dyan. Okay, so that makes the presence of nitrogen with the carbon atom here, of course, in the vertices of this hexagon. Okay, dito may mga carbon atom dyan. With this presence of carbon atom and nitrogen in the cyclic formation, yan yung sinasabi nating heterocyclic kasi nga, hindi siya purely carbon atom. Okay, and may nitrogen siya. Okay, so there are five heterocyclic bases that could be found in the nucleotide. And three of them are, so we have two classification. We have pyrimidine and purine. Okay, three of that five nitrogen bases are pyrimidine, and two out of five bases are purine. Okay? Pag sa structure naman ng ating pyrimidine, it is a monocyclic base. So, isa lang siya, isang cyclic formation lang siya for that pyrimidine. And there are three specific bases. These are thymine, cytosine, and uracil. These bases could be abbreviated by one letter. Okay, so meron silang one letter abbreviation. Thymine is T, cytosine is C, and uracil is U. Please do not get confused dun sa names ng ating amino acid at saka ng ating bases. Kasi meron din tayo, remember dun sa ating proteins, meron din tayong T, okay, na tinatawag dun sa ating amino acids. But pag pinag-uusapan natin ang nucleic acids at nakakita tayo ng T, of course, that represents thymine. Okay? And then, cytosine C and uracil is U. So, makikita natin dito that it is only composed of one cyclic or cyclic structure. Kaya monocyclic ang pyrimidine. So, if you're going to compare pyrimidine versus purine, which is a bicyclic, okay, so tinangnan natin yung ating purine. Okay, ito. Meron siyang dalawang cyclic components. So, this one is a five-membered kasi di ba pentagon yan. Tapos ito naman yung six-membered ring. Okay, so yan yung sinasabi niya dito kanina. Purine is a bicyclic bases fused with 5 and 6-membered rings. Okay. So if you have to compare the molecular weight of this purine and pyrimidine, mas mabigat or mas malaking compound ang purine because of that bicyclic structure, and purine has smaller structure. Okay? And we have two purine derivatives, namely adenine and guanine. Okay? Adenine is denoted by letter A and guanine is denoted by letter G. Again, ulitin natin, pag nakakita kayo ng G, parang ito G. Kasi dun sa amino acid, meron tayong G, glycine. Pero pag G na nucleic acid or bases rather, at nagamit siya sa nucleic acid, of course, it represents guanine. Okay? Now, okay. For these bases, let's all remember that adenine, guanine, and cytosine are common or they could be found for both RNA and DNA. While uracil is unique only for RNA, so exclusive lang siya kay RNA, and thymine is exclusive only for the DNA. Okay, so with this structure, with this uracil and thymine being unique to RNA and DNA respectively, makikita mo pa lang dun sa sequence kung RNA sequence ba siya or DNA sequence. Okay? Now, for the third and last component of the nucleotide, we have the phosphate or HPO4 negative 2. This is the last or the third component of the nucleotide, which is actually derived from phosphoric acid, which is H3PO4. So, ito yung phosphoric acid. It could actually be dissociated into hydrogen phosphate ion. Okay? Now, how are nucleotides being formed? Okay, so for the nucleotide formation, we have some important characteristics here. Okay, so The formation of nucleotide, of course, with that three components, they could be formed via condensation, which is the formation of water molecules. And there are two locations for this condensation reaction. It is between sugar and base, and the second one would be between sugar and phosphate. Kaya, tignan natin dito. This is the condensation na sinasabi. So, this is the sugar, and this is the base, and this is the phosphate. Okay, so upon the reaction of phosphate to sugar, there's a condensation reaction wherein the phosphate will be bonded to the fifth carbon atom of the sugar and the production of water. It is sinasabi niya production of water kasi the hydroxyl coming from the phosphate and the hydrogen coming from the sugar will be combined together, and thus the formation of water. And the second reaction would be between the second condensation reaction occurs between the base and the sugar again. So for this case, the nitrogen here, the hydrogen coming from this base and the hydroxyl will react together to form the base, and hence formation of bond between the base and the sugar will be formed. So ito na, magiging as one na sila. Okay? So again, the phosphate has been attached to the fifth carbon on the pentose sugar, and the nitrogen coming from the base would be attached to carbon number 1. So ito naman of the carbon number 1 or 1 prime of the pentose sugar. So that's the condensation reaction. So meron tayong formation of water molecules kapag nag-combine yung mga components ng ating nucleotide. For the second characteristics, so ito yung sabi ko kanina, the base is always attached to carbon 1 prime of the sugar. So ito yon. So, ito yung carbon number 1. So, dyan siya ma-attach ang ating base. While as for the phosphate, nasa 5' siya. Okay? So, for purine bases, if that's a purine, ang attachment daw niya sa sugar ay through N9. And while as for pyrimidine, nasa N1 siya. Okay? So, kapag titingnan natin, ito yung ating N9. Yan yung N9 natin. Okay? Because that is for purine. Pagdating sa ating, sabi niya dito, for pyrimidine, it is attached to that C1 of the sugar and N1. So, hanap tayo ng example dito sa ating notes. So, ito yung N1 naman. Okay? So, this is the carbon 1 prime of the sugar, and the bases, ito naman yung N1 niya. Tapos, still, this is carbon number 1, pero this one naman yung N9 ng purine. Ito yung 9, tapos ito naman yung sa 1, N1 ng pyrimidine. Okay? So, ulitin natin, the bases was attached to carbon number 1 of the sugar, and depende kung anong base yan. If that's purine bases, ang attachment is N1 to C1. If that's purine, N9 to C1, and then for pyrimidine, N1 to C1. Okay? So, and the bonding would be a beta and glycosidic linkage. Beta configuration, so recall lang natin ano ba yung beta configuration pagdating sa mga sugar. It could be the components and the carbon 1 and the fifth carbon atom should be on the same side. Okay, so same side siya. Okay, so ito nasa taas din siya. Kapag alpha kasi, remember for alpha, yung isa nasa taas, yung isa dapat nandito sa baba. Okay? But for the orientation of this nucleotide, palaging beta orientation. Parehas sila on the same side. Nasa up part siya. And of course, the phosphate group, ito yung sabi natin kanina, the phosphate group has been attached to the sugar at carbon 5'. Ito naman yung carbon 5'. Through a phosphate ester linkage naman. Okay? Now, we have 8 nucleotides, okay, that are building blocks for DNA and RNA. 4 for DNA, 4, and another 4 for RNA. Okay? So, we have here, of course, madidistinguish mo kung DNA siya kasi nga, meron siyang, sa nucleotide name niya, merong deoxy. Okay? So, we have here the bases, adenine, guanine, and cytosine, both for RNA and DNA. Okay, so we have thymine for DNA and uracil for RNA. Okay. And then this is now the primary nucleic acid structure. So this one would be nucleotide 1, and then another group will be nucleotide 2 and nucleotide 3. Remember, the building blocks of nucleic acids are nucleotide. Okay, so it has a 5' end and 3' end. So ang pagbasa natin is from 5' to 3'. So again, the phosphate and the sugar is attached together via phosphate ester linkage, while as the base has been attached to sugar via glycosidic bond. Okay. Meron din tayong tinatawag na a non-terminal phosphate. Ito yung sabi niya dito. Each non-terminal phosphate in the nucleic acid backbone is bonded through a 3' to 2 sugars. Ito yon, 3', 5' phosphodiester linkage. Kung kanina, sabi natin, the terminal phosphate, pag sinabing terminal yung nasa dulo, terminal phosphate is bonded to this sugar via phosphate ester linkage. However, yung ating mga phosphate non-terminal, yung mga nasa gitna, they are bonded to sugar via 3', 5' phosphodiester linkage. Okay? And of course, yung sabi nating pagbasa kanina from 5' to 3', and we call that directionality. Okay? So, each chain will have a directionality such that we are going to read the bases from 5' to 3' end. Kasi bases lang naman ang magbabago dito. Consistent pa rin naman na may phosphate sugar kapag DNA, phosphate deoxyribose, phosphate deoxyribose lang. Pag RNA, phosphate ribose lang ang ating backbone. Backbone ang tawag dyan sa repeating units ng phosphate sugar and phosphate sugar. Kaya pag binasa natin, ang tinitingnan lang natin is the identity of the bases. Kaya puro letters na AUG, ang makikita natin. Okay? So, meron tayong tinatawag na nucleic acid backbone. This is the alternating sugar phosphate chain in the nucleus. So, ito yung ating backbone. Okay? So, for RNA naman, so RNA, sabi na, ganun pa rin, meron siyang phosphate sugar, but this time this is the ribose. So, ang mga heterocyclic bases niya is the adenine, cytosine, guanine, and uracil. Still, sugar has been attached to ribose via glycosidic linkage. Phosphate to ribose is through phosphate ester linkage, and the phosphate in between, or the non-terminal phosphate, is attached to the sugar via 3', 5' phosphodiester linkage. Okay, so directionality nito, okay, is read from the 5' to 3', and then we are just representing, or we are just going to name the name of the bases. Okay, so for RNA, an example nito ay 5' CGAU 3'. Okay, so ito yung ating CGA, okay, the CGAU. Ito na yung kanyang backbone. Alternating phosphate, sugar, phosphate, sugar. Okay, so this is the 5' end, and dito sa baba yung 3' end. Okay, so makikita natin that malaki talaga yung molecules ng ating mga purine, and itong pyrimidine natin ay small structure lang. Okay, still, This bases has been attached to carbon number 1 prime in the pentose, and the phosphate is on carbon number 5. Okay? So, for DNA naman, ganun pa rin. We have here the phosphate and the sugar, which is the deoxyribose naman this time. And the bases are adenine pa rin, cytosine, and guanine, but the unique is the thymine. Okay, so an example of a DNA structure, meron siyang T, kasi yan ang kanyang identity or unique sa kanya. Still ang pagbasa, 5' T, G, C, A, then 3'. Okay, so this is the thymine. The N1 is been attached to carbon number 1, and then for guanine, N9 is attached to carbon number 1 of the phosphate. Sorry, of the sugar. And the phosphate is attached to carbon number 5. Okay? So those are an example of DNA and RNA structure. Now, if we're going to compare the nucleic acid versus protein, okay? So we have here the nucleic acid has 5' and 5' end and 3' end. While as yung protein naman has N-terminal and C-terminal. So tingnan natin yung structure. So this is a nucleic acid. It has a 5' end. Okay, while as yung protein naman meron siyang N-terminal. Okay, and ito naman yung C-terminal. Okay, ito yung C-terminal. Okay, and the nucleic acid has nitrogen bases. Ito yan, base 1 na naka-attach sa kanyang backbone. While as for proteins, yung R, amino acid residue. Okay. Both nucleic acid has backbones. Sa backbone ng nucleic acid ay alternating phosphate and sugar. While as for amino acid, we have here the alpha carbon atom and the peptide bond. So, alpha carbon atom and peptide bond ang kanyang backbone or alternating unit. Both nucleic acid and protein has directionality. Okay, so binabasa natin ang nucleic acid from 5' to 3', while as for proteins, we read it from N-terminal to C-terminal, parang pa left to right. And then for nucleic acids, like nucleic acids, protein also has the primary and secondary structure. Okay? For the DNA, okay, let's all be reminded that DNA has double helix, while as for RNA, single strand lang siya. Okay? So, what's unique for DNA double helix? So, it involves two polynucleotide strands. Ito, meron siyang isang strand going from 5' to 3', and meron din siyang second strand going from 3' to 5'. And of course, these two strands form a spiral staircase. Parang ang itsura niya, helical kasi siya eh. So, it is coiled in a manner na parang spiral staircase siya. Okay, so yung hagdan na parang pa-spiral. Okay, and the backbone, the sugar phosphate backbone of these two polynucleotide can be thought as being the outside banister of the spiral staircase. So, ito daw yun. Parang yan ang kanyang outside banister. And then the bases naman are extended inward. So hindi palabas, rather paloob kasi meron silang attraction. The bases from the first strand will have an attraction through hydrogen bonding dun sa bases dun sa second strand. Okay? And of course, these two strands are anti-parallel. Pag sinabi nating anti-parallel, one strand will run from 5' to 3' direction, and the second strand will be 3' to 5'. So, baliktad. Magkabaliktad siya. So, ito yun. Okay. So, dito sa itong blue strand na to, mag-start siya sa 5', tapos mag-e-end naman siya sa 3'. Ito. Tapos, yung ating kanyang second strand naman, dito ay 3' naman. And then, mag-e-end siya dito ng 5'. Okay? So, we could see Itong parang ribbon na ito, this one would be the backbone. It is composed of the sugar and phosphate repeating unit and then or alternating unit. Tapos dito pa loob naman yung ating mga bases. Okay? At yung attraction nito, hydrogen bonding, is we call them as base pairing. Okay? Always bear in mind that adenine is paired with thymine and cytosine is always paired with guanine. Okay? So, for the base pairing, the relative amount of this base pairing varies depending on the life form from which the DNA is obtained. Each animal or plant has a unique base composition. However, the relationship of A to T is always equal. Okay, so the percentage of A is equal to percentage of the T. Okay? So, si adenine, ang percentage ng adenine sa ating DNA ay palaging equal sa percentage ng thymine. Ganon din si cytosine tsaka si guanine. Okay? So, bakit paanong naging parehas sila ng percentage? Kasi nga, if we're going to have the double helix structure, so kunyari meron tayong A dito, may dalawang A, okay? So, pagdating dun sa second strand, so syempre meron tayong T dyan. So, kung ilan yung percentage ng A, ganun din yung percentage ng T. Okay, kung meron naman dito na C, ganun din naman, magbe-base pair siya ng G. Kaya, pag DNA, percentage of adenine is equal to percentage of thymine. Dahil yun sa double helix structure niya. And base pairing, of course. Now, for physical restrictions, the size of the interior of size of the interior of the DNA double helix limits the base pairing. Kasi, remember that we have two purine and two pyrimidine and two purine, di ba? Sa ating DNA. However, bakit hindi pwedeng mag-pairs yung dalawang pyrimidine or mag-pair yung dalawang purine? Okay? So, ito yung reason bakit si A at saka AT at saka GC lang yung ating favorable. It is because that pyrimidine, okay, so remember monocyclic lang si pyrimidine. So kapag dalawang pyrimidine yon ang mag-base pair, sabi dito, very small ang kanyang structure. While as kapag dalawang purine naman, remember that purine are bicyclic. So medyo malaking masyado na yung ating structure. So dun sa loob ng ating helical interior or helical structure, hindi na magkakasya yung dalawang bicyclic structure ng purine. Pag pyrimidine naman, monocyclic, malaki naman yung space. So, ang ideal daw is one purine tsaka one pyrimidine ang dapat. Okay, so sabi niya dito, there is not enough room for two large purine kasi nga bicyclic siya or yung two small pyrimidine naman very too far, so madami naman siyang spaces. So, possible lang is combination ng purine and pyrimidine. But for this combination ng purine and pyrimidine, meron tayong 4 possible combinations. Pwede daw na mag-combine si A and T, okay, so adenine and thymine, and this base pairing will exhibit 2 hydrogen bond. Pag si A naman, adenine, nag-pair siya kay cytosine, meron siyang 1 hydrogen bond. Si guanine naman and thymine, 1 hydrogen bond din. And then for guanine and cytosine, may 3 hydrogen bond. Okay. Most favorable ang combination ng A and T dahil sa dami ng hydrogen bond. Si A and T meron siyang 2. Si GC, meron siyang 3. Hindi na favored si AC kasi very weak. Tag 1 hydrogen lang siya. So, pagdating sa DNA, dalawa lang ang ating favorable base pair. Yun ay CAT pairing and GC pairing. Okay. But let's all remember that pag sinabi natin that the attraction between hydrogen bonds, relatively weak lang yan. Okay? Mas malakas pa rin yung mga ionic bond like that. But kahit na mahina ang hydrogen bonding natin, pero dahil sa sobrang dami ng hydrogen, lumalakas pa rin. That's why it's significant. Meron pa rin siyang significant strength. Okay? So, ulitin natin. The base pairing between purine and pyrimidine, hindi favorable ang dalawang purine kasi masyadong maliit. Pag dalawang purine naman, hindi siya favorable kasi masyadong malaki. So, ang idea lang is 1 pyrimidine and 1 purine. Okay? Pero, dito sa 1 pyrimidine and 1 purine, meron na namang favorable. Yung AT and GC pairing. Hindi favorable si AC kasi nga mahina yung 1 hydrogen bond niya. Tapos, hindi rin favorable si GT kasi ganun din. 1 hydrogen din. 1 hydrogen bond. So, mahina. Okay? For complementary bases, the third characteristics, the complementary bases, pag sinabi natin complementary bases, ito na yung base pair niya. So, yun, kanina na-discuss natin, A always pairs with T, and G always pairs with C. Okay, so adenine and thymine are complementary. Yun yung term ha, complementary. Okay, and guanine and cytosine are complementary. Okay. Now, since we have two strands in the double helix structure, we have two strands in the DNA in double helix, these strands are not identical. Kasi nga, kung yung isang strand meron siyang A, T, tapos yung second strand naman, okay, of course, meron siyang, okay, sorry, dagdagan natin tong isa. Itong first strand may G, C, or GG, okay? Yung sa second strand, of course, meron na tayong T, tas A, tapos ito naman ay C. Sorry. Ito naman ay C. C. Okay? So, hindi siya identical kasi ito, yung first strand is AT, GG, tapos yung second strand naman ay TA, CC. Okay? So, magkaiba. Hindi sila identical. Rather, ang tawag sa kanila ay complementary because of the base pairing. Okay? Now, if the end designation, yung 5' or 3' end is not specified, always, kunyari, ang binigay lang sa'yo is A, T, G, C, ganyan lang binigay. Always remember, kapag walang 5', 3' end, always assume that it starts with the 5' and ends with the 3'. Okay? Now, for the complementary strand that runs in 3' to 5' direction, kasi nga anti-parallel siya, okay? So, pwede rin this is also acceptable kasi nga anti-parallel siya. But you could actually reverse it. So, yung 3' to 5' could be converted to 5' to 3' by reversing the order. Okay, makikita natin mamaya kung paano tayo magre-reverse. Okay? And of course, ito yung sabi natin kanina, the strands or the bases in the base sequence of the that could be found in the strand of DNA or RNA, we list them using the one-letter abbreviation. Okay? So, let's have this example. So, if we're going to have the given strand, malalaman natin na DNA ito dahil merong T. Okay? Because that's unique for DNA. So, if we're going to have the given strand at required tayo to write the complementary, of course, ang step by is, if we're going to have 5', of course, anti-parallel, lalagay natin 3'. And then, always bear in mind, yung ating base pairing at GC palagi. Okay? So, adenine pairs with thymine and guanine pairs with cytosine. So, at GC. So if we're going to have here C, then of course base pair niya si G. G base pair with C, and A with T, T with A, C with G, T with A, and A with T. And of course, pag yung 3' naman, ang anti-parallel naman niya ay 5'. Okay? So this is now the complementary of the given structure. Now, sabi natin kanina, anti-parallel, di ba binabasa natin ng 5' to 3'. Pero yung complementary, hindi siya nakasulat sa correct directionality. So, pwede naman natin siyang reverse. So, ito, mag-start tayo sa 5', tapos susunod niyan ay T, and then we have A. So, i-reverse lang natin. And then we have G, G, A, tapos itong T na dalawa, and then CG. CG. And then, of course, we have the 3'. Okay? So, the complementary could be written from 3' to 5', pero pwede rin natin siyang i-reverse. So, pag tinanong tayo, is the given 5', given yan, tsaka ito, mag-complementary, yes. Pag mamatchan mo lang siya, 3' to 5' to 3'. Okay? So, this one again, 5'. So, ang anti-parallel niya, 3'. The A, base pairs with T. Ito rin. Of course, the T with A. GC. Ito, CG. AT. Okay. GC. CGTA. And of course, dun sa last, 3, pag may 3', of course, susulat natin yung 5'. Pag i-rewrite natin ito, complementary strand niya, mag-start tayo sa 5', 5', and then we have AG, AG, tapos susunod itong C, and then we have T, G, C, A, and then we have the T, T, and then, of course, the last is 3'. Okay? So, this strand still a complementary of the given.